things in week 3. We got some career path interviews. We're going to talk with Dr. Brooke Topoisomerase. We're going to talk with Scott Sewell and some other folks. We also got a meeting with our ancient DNA collaborators. Actually one on Monday and another meeting on Thursday. So we're excited about those, making great progress on several fronts. So the ancient DNA work continues. We're hopefully going to take our base model to a place where we can actually see the data, the output piece of it. Kind of visualizing it is really important. The topoisomerase work, we're going to start purifying mutants. So we've got several that are harvested and ready to go. We just need to get those purified and then be able to run experiments on them and then start growing the last of them. So work our way through that throughout the week. Going to try some new experiments this week. So we've got a new binding assay we're going to try out. We're excited about that. We're continuing to write up some of Xavier's work. And so we're just chipping away at the little pieces along the way. This is a new flow cell, so you can see the things that got to go into the unit there. So you're going to stick it in with this end, going up under the clip. Nanopore 4S says it's got almost 1,700 pores, so that's really good. So it's a good, clean flow cell ready to go. This flow cell flush that hole, slowly overflow chamber filling up a little bit. Your sample. What you want to do here is you want to get down here, but you want to do it kind of dropwise. So you see how it kind of wicks its way in there? Mm-hmm. There's a little capillary action going on. Close this. And close this. These are.
See if we have any like topo in our yeast growth. So we have our growth.
Yeah.
So when we image this, we should see a very clear band at the top.
Yeah.
And if we don't see that, that means there's no topo. So far we have seen no topo.
Well, we just finished looking at different mutants under the microscope, and basically what we found is that certain mutants weren't growing and had a lot of bacteria in it, or just weren't growing in general. So what we're doing now is we're running a protein genome to see if topo is present or not, and we're also getting rid of a couple mutants and starting over. But some of them look good. Look promising. So we'll see what happens.
Visually. Conversation filling the blue is nickel atoms that are bonded to this resin. So that's what's going to capture our topo. Mixing things. This is like space age.
Yeah.
Yep. And I turned it on. We got this closed. We'll put our sample in here. Next we need to put our column in. Then once it starts loading, it'll switch down.
A little different stuff today. Right now we're making what's called a P cell. Basically, it's what's going to be catching the topoisomerase as we drip it through. The only problem is this pH is supposed to be like 7 and it's like up to 9 right now, which is not good. We do not want that. So right now we're making something a little bit more acidic to help bring that down.
We don't know what the ends of this DNA looks like. And basically what we're trying to do is just clean them up so that they have ends that we can attach the barcodes to and then attach the native adapter to that. We're going to prep this, and then once we're done with that, we'll load 200 more of the prime mutant. I'll let you kind of tell of actually using light to detect fluorophores or fluorescent plate like this, and they'll have a fluorophore that will incorporate into the DNA the more it gets copied. And so the more product that's formed, the more fluorescence will be present in your sample, the more it will get reported in the plate reading. Does that kind of make sense? So we call that quantitative PCR. You might also hear it called real-time PCR, so RT-PCR or qPCR. RT-PCR can also stand for reverse transcriptase PCR. So if you have RNA, you can convert the RNA to DNA and then run PCR in this machine and see. That would be a way to measure, like, I want to know, is gene A expressed more than gene B? Well, you could run RT-PCR convert the messenger RNA of those genes to a DNA sequence, amplify it in here, and see which one is higher under. This is the best part. Y'all ready for this? I gotta see your
Y'all,
space age.
it moves.
Here we are, 2026, summer research t-shirt.
Answer to that. Internally, I'm very careful to partition my data so that I know that I'm only using my data inside.
I'm doing another internship with a company.
Model to actually identify uracil in DNA data.
I can't even add them here, and they'll show up. And if I zoom in close enough to these, then you can.
Poured some liquid nitrogen in here, so it's a little cold. And it went from this kind of liquidy stuff to now grind it up into a little powder.
And I was like,
That's how.
I got freezer burn the other day.
Oh my goodness, my fingers are cool.
pretty cool. This thing has been giving us issues because it doesn't spin. So we kind
Sequencing is
You're not using
working.
your own.
Quality control check. Epitome is a program from Oxford Nanopore Technology that allows you to run specific analyses of your sequencing data. And in today's case, we're looking at Dr. Kersey's bacterial genome data, and we want to know, can it assemble the whole genome from all the fragments? And so if you can picture, we sliced this genome up. It's 5 million base pairs, 5 million letters long. And we sliced it up into little fragments, sequenced it, and now we got to go back and actually assemble those into a continuous sequence because bacterial chromosomes are a circle.
we are not there yet. We did a lot of practice runs, like on the different assays in the very beginning of the program. This week it's been a lot of learning, a lot of trial and error. But overall, we were able to kind of see the mutants that were variations of topo that may not look too good, and then that look better. And so we were kind of able to like say, okay, we're going to focus on this and not on that.
Gotcha.
Seeing what's worth it, what's not. Ran out of protein gels. Crushed a lot of.
This week has been learning the process of getting our topo purified, getting it ready to be tested. So it's a lot that we're learning. And yes, there's been a lot of failure this week. We've had to redo things. We've had to try out new methods, troubleshoot, MacGyver stuff. But ultimately next week we should have a good standard. We should know what we're doing. We should know what we're working with, and we should be able to come back a lot stronger with the new meetings. So hopefully, we're ending this week a little down, but next week we are hopeful that things will look better.
And I mean, we got to practice a lot of skills too.
Yeah.
We did a lot of, well, as far as like identifying stuff under a microscope, and then, like I said, a lot of protein gels. We didn't really do that a lot in the beginning of the program, so now we're a lot more familiar, seeing what works and what doesn't, what's a little bit better for us or for each other. So
I've learned
pretty good.
the computer system a lot more clearly, like how to troubleshoot on there, connected to the machine, everything with the prep.
Okay. Hey guys, so we've reached the end of the week. I'm with my partner, she's matching with me, and we are super excited for next week that's coming because we're sequencing more samples. We have sampled a couple of DNA, and so next week we'll probably have more. So we want to transfer that information to the bioinformatics team. So we're looking forward to what information we get from that. And the more the merrier. You know, we are not looking forward to getting everything wrapped up this whole summer, but at least we're learning many more skill sets, and that's very great for us. Kenna, do you have anything to say for next week?
I'm excited to get started on next week because we've done a lot of the preparation stuff, and we've gone through sequence things, but we get to actually apply what we got from the sequences next week. So I'm really excited about that.
All right, stay tuned, guys, and we are reaching the end of the summer, so stay locked in.
Kicking the students out, sending them off. It's been a busy week. On the bioinformatics front, we continue to work on this base calling model. The new challenge is visualization. How can we see what the data is showing us? And that is still an ongoing question. We've tested our new base calling model on some different data sets, and it is consistent with our expectations, but we're still learning about it. So there's still a lot to go on understanding the model, understanding can we improve it, but the big victory there is that it seems to be working, and we're super excited about that. So we're inching closer on that. We had some great meetings with collaborators this week, and so we're super excited about that. On the ancient DNA sequencing front, we got some new samples sequenced. We haven't even processed that data yet, like it's still sitting there. So excited about that, but at some point we got to get to it and start working with that data. And we got some more samples that, Lord willing, we'll sequence next week, so maybe even on Monday. On the topo front, we've been stymied. So we've had great progress on these other sides. This one we've been just kind of stuck. And the issue was contamination with some of our yeast lines, and so just trying to get all that cleaned up, and now purification. So getting the cells growing, getting the protein expressed, and then getting it purified so then we can study it. And today we tried to do another purification. We had a hiccup, and we might have got something, but we're gonna have to wait till Monday and see if we can clean it up and get something out of it. So little by little, we're going to get there, but it's just it's a slow go, and so we're just having to be patient and trust that we can get there a step at a time as we keep moving forward. So thanks for being out there, and continue to pray for our work.